Hi, I'm Hiromi Tsuruta from Cognano. I'm very excited to tell you about the work on a large-scale dataset for therapeutic antibody discovery. Let me start with a brief introduction of our background. Nowadays, antibodies have become an important class of therapeutic agents to treat human diseases because of their high target specificity and binding affinity. In the development of therapeutic antibodies, it's important to identify specific interactions between antibody candidates and target antigens, called antigen-antibody interactions. So to accelerate therapeutic antibody discovery, machine learning has attracted interest for predicting antigen-antibody interactions. Currently, there are some public datasets related to antigen-antibody interactions, as listed in this table. Although these are very useful datasets, they have some notable limitations, such as small sample size, the lack of non-binding samples, and the lack of exact amino acid sequences of the antigens. To overcome these limitations and facilitate machine learning-based antibody discovery, we release a high-quality, large-scale dataset for predicting antigen-antibody interactions. In our research, we established a novel data generation method using the immune system of a live alpaca. The reason why we use alpaca is that alpacas are known to have simple structure antibodies called VHH, and their amino acid sequence can be identified more easily than conventional antibodies. In our data generation process, we first immunize an alpaca with a human IL-6 protein that we use as a target antigen. After immunization, lymph nodes and blood samples were collected from the alpaca's body. Then we performed an affinity selection to distinguish between VHHs that bind to IL-6 protein and those that do not. The resulting dataset contains the amino acid sequences of the IL-6 protein and the VHHs and the binary levels for binder or non-binder. Importantly, as antigens, we use the wild-type IL-6 protein and its 30 mutants produced by artificial point mutations, and our dataset contains the binding information of diverse VHHs to each antigen mutant. As the COVID-19 pandemic has shown, viruses continuously evolve through mutation. Because such mutation can lead to profound changes in antibody binding, prediction of their effect is important in the development of therapeutic antibodies. This figure visualizes whether randomly selected VHHs bind to each antigen type or not. As a result, our dataset contains many sensitive cases in which point mutations enhance or inhibit antibody binding, providing variable benchmarks for predicting the effect of antigen mutations on antibody binding. And we reported benchmark results on our dataset by using machine learning models. Our experimental scenario assumes that antigen mutants emerged one after another, and we want to predict antibody candidates that will bind to future emerging mutants according to the binding information that has already been observed. To do so, we randomly split the dataset into training and test sets based on antigen types. Then we trained the model by using only the wild-type IL-6 protein and evaluated their performances for unknown mutants contained only the test set. Next, we randomly select one mutant from the training set and added it to the model's training data to evaluate the model performance. By repeating this process, we track the model's predictive performance for the test set. These figures show the precision, recall, and F1 score of the baseline models as a function of the number of IL-6 protein types used for model training. All the metrics improve as the number of IL-6 protein types increase, but after reaching saturation, the recall were still only about 85%, even for the 3 neural network-based models, indicating that the models failed to predict about 15% of the binding antibodies. So further research on model architectures for predicting antigen-antibody interactions is needed, and our dataset will be a useful benchmark for evaluating such models. In the future, we plan to generate and release datasets for various antigens to establish a more comprehensive database of antigen-antibody interactions. Thank you for your attention.